We make decisions every day, like what to eat and where to go. Sometimes we make good decisions, and sometimes we make bad decisions. Would it be helpful to better understand how our brains do this? To be able to understand how the brain makes decisions, we use model organisms to test different types of experiments to understand how these animals make decisions. And one of them is a fruit fly. What makes these flies so important for science? First of all, they reproduce quickly. Their life cycle only lasts 10 days. Second, they're easy to manage. They're very small. As you can see in comparison to the mouse brain, the Drosophila fly is very small. And third, the complete genome sequence of Drosophila was published in the 2000, making it easier for us to study them now. Now that we know the importance of flies, let's study their behavior under different scenarios. When we try to understand behavior, we often look at genes that alter the behavior of an animal. The reason when we study their behavior is because 60% of their genome is homologous to human and 75% of the genes known to be responsible for human diseases. We hope to increase our understanding of the human genome by observing the behavior of these organisms by manipulating certain genes. We're going to be focusing on the behavior of chemiosensation in Drosophila melanogaster larvae under 3 different scenarios: the caffeine barrier, the yeast, and the combined. This is an example on how the Petri plates are going to be organized under the 3 different scenarios. Chemiosensation behavior is one of the most important coping mechanisms for the living organisms, including humans, to survive in the environment, helping us sense the chemical reactions of the world. This behavior is analyzed by observing the willingness of larvae to take a risk by moving over an aversive chemical stimulus to reach food or reward. The normal behavior of Drosophila melanogaster larvae in their interstages, all their energy is focused on acquiring nutrients known as a foraging behavior. Will this foraging behavior be enough to push them to cross the chemical barrier? We analyzed the larva preface of the plate under the different scenarios dividing the plate in different regions. We started with the yeast plate, the orange representing the middle region, blue the end, and black the start. The same with the caffeine. And lastly, the combined, the yeast and caffeine scenario with the same regions. This is an example video of an experiment with yeast and caffeine on the plate. We record this video in order to quantify the number of larvae and where they are on the plate every 30 seconds. Both yeast and caffeine are at the end point. Even these larvae are genetically very similar to each other, they can behave very differently in the same environment. Some larvae crawl straight to the food source, ignoring the caffeine barrier. Other larvae crawl towards the food source and then turn around upon reaching the caffeine barrier. Then we quantified our results making 3 graphs of each specific scenario. In the x-axis shows the time during the trial in minutes, and the y-axis represents the percentage of larvae in a specific region. The colors represent each region, with the start being gray, the middle orange, and the end region with the caffeine and or the yeast in blue. In the first scenario of yeast only, the larva percentage of the start region decreased throughout the trial, as the percentage in the middle and end region increased, representing the larva pursuing the smell of the yeast. The caffeine scenario shows an increase in the number of larvae in the end region from 2 to 3 minutes, meaning that the larvae explore the plate. Intrinsically, the number of larvae in the end region decreased in the last minute, representing the moving away from the aversive caffeine region. And the combined scenario, even with the aversive barrier of caffeine, many of the larvae choose the risk of crossing the barrier to reach the yeast, and they stay throughout the trial. This represents their willingness to tolerate the unpleasant caffeine in order to find the yeast reward. Here are the final results after collecting more data, around 4 to 6 experiments per condition, On the x-axis, we have each of the three scenarios, and on the y-axis, the percent of larvae in the end region after 4 minutes. We found that the number of larvae that reach the endpoint is highest in the yeast-only condition, demonstrating that the larvae are attracted to the smell of yeast. The number of larvae that reach the endpoint is lowest in the caffeine-only condition, demonstrating that the larvae are averse to caffeine. Finally, when combining the caffeine and yeast on the plate, the number of larvae that reach the endpoint is intermediate. Although the effects were not satisfyingly significant, is determined by a one-way ANOVA with Donnett's multiple comparison test, we believe it will be significant after conducting more experiments. These findings shows that larvae can detect stimuli such as smell and taste, and make decisions about whether or not to search for food despite an aversive barrier. In conclusion, after analyzing our data, the Drosophila melanogaster larvae chose to take the risk of crossing the chemiosensory barrier to obtain the reward, starting at the start region and exploring the rest of the regions of the plate, demonstrating that the smell of food increased their willingness to take risk in their foraging behavior. In the scenarios with the caffeine barrier present, they preferred to stay at the start region, which demonstrates that they can sense the caffeine barrier and find it to be aversive. In contrast, the caffeine wasn't present most of the time they were in the middle. The preliminary experiments provide evidence that the larvae can be used to start the risk-reward decision-making. In future directions, future experiments will involve the activation and silencing of specific neurons, such as dopaminergic neurons, to see how they influence their decision-making behavior to better understand the neural circuits underlying decision-making, making it possible to understand better the mental health problems to develop better treatments for this field.